Eileen, you are up. So Eileen is a senior at Amador and has joined the group about 2 years ago. Initially as a very awkward and shy student, but now less awkward, less shy, and more interested in organic synthesis and its applications in drug discovery. So Eileen will be sharing the latest and greatest in this project that's been going on for quite a while, which we hope to hopefully have a paper out pretty soon. So check it out.
Can you guys hear me? Can you hear me?
Yes.
Okay.
Yes.
Hello, everyone. My name is Eileen. I'm a rising senior at Amador Valley High School, and I'm part of Dr. News Group. Today, I'll be talking about my project, which is the design and synthesis of novel Carmofur analogs, as well as the in vitro evaluation against cancer cells and the SARS-CoV-2 main protease that we did. I'll start out by talking about an introduction and background to Carmofur. Secondly, I will talk about the overview of my project and the goals. Thirdly, I will talk about the analog design and synthesis. And lastly, I'll cover the in vitro studies that we did with the molecules that we made. So a little bit of background. Ceramide is a pro-apoptotic agent. So it's vital in carrying out normal programmed cell death, also known as apoptosis. Acid-ceramidase is an enzyme that is responsible for degrading ceramide into sphingosine and fatty acid, which are anti-apoptotic. When acid-ceramidase is overexpressed, it reduces ceramide-induced apoptosis, which can then lead to cancer. In fact, 40% of primary prostate cancer tissues demonstrate acid-ceramidase overexpression. Therefore, by inhibiting acid-ceramidase, ceramide breakdown would be reduced, and that would inhibit the growth of cancer tumors and maintain normal apoptosis levels. One compound that inhibits acid-ceramidase is Carmofur. It was initially developed in 1981 in Japan as a treatment of colorectal cancer. By inhibiting acid-ceramidase, cancer progression is limited because there would be ceramide-induced apoptosis. Now let's switch gears and look at drug repurposing and why it's beneficial. Drug repurposing is basically taking existing FDA-approved drugs and repurposing them for a different disease. And there's a lot of benefits to us. For example, the average number of years from initial development to regulatory approval of new drugs is anywhere between 10 to 12 years for one drug, which is a long time. However, this number goes down to 4 years for repurposed drugs. In addition, repurposed drugs have a much higher success rate than new drugs. Some examples of successfully repurposed drugs include zidovudine, which was originally designed to target cancer and was repurposed for HIV and AIDS. Fingolimod, which was originally used for transplant rejection and was repurposed for multiple sclerosis. And lastly, topiramate, which was designed for epilepsy, but repurposed for obesity. During COVID-19, drug repurposing is what the researchers, is the method that the researchers used because they wanted to develop a treatment for COVID-19 as efficiently and with as much success as possible. So through a high-throughput drug repurposing screen, Carmofur was identified as an inhibitor of the SARS-CoV-2 main protease. And so it was a promising drug that could be repurposed to treat COVID-19. Now, what exactly is the SARS-CoV-2 main protease and why is it important in COVID-19? Polypeptides required for viral replication of SARS-CoV-2 are released predominantly through the main protease or the Mpro protein, which plays a major role in the replicase transcriptase complex. Its crucial role in viral replication across many variations of coronavirus make it a promising target for anti-COVID treatment, because if we can inhibit Mpro activity, then viral replication would be restricted and thereby stopping the spread of the virus in the body. So what our group previously worked on was we used 19F NMR to optimize the synthesis of Carmofur, because previously Carmofur the synthesis required using pyridine, which is a toxic solvent. It required a reaction time of 72 hours with less than 35% yield. So by using 19F NMR to optimize the synthesis, we were able to switch to a non-toxic solvent, a less toxic solvent, NMP, and we were able to reduce the reaction time drastically. In addition, we ended up with over 70% yield, which is much better. After we found a successful optimization of Carmofur, we wondered if we can apply this rapid reaction workflow for the preparation of Carmofur analogs, which may perform better than Carmofur. So we designed a library of 10 urethane and single atom substituted analogs of Carmofur, with each group designed to target different characteristics and properties of biological activity. We designed the benzyl urethane analog to target the aromatic residues in Mpro. And the image that you see on the right is from computational docking. So we basically docked the molecule to Mpro, and the number negative 6.4 kcal per mole is the docking score. So the more negative this number is, the better the binding affinity. Additionally, we want to design an analog with a cyclized chain to probe the effects that such a substructure may have on binding affinity. So we designed the transfer methyl cyclohexyl urethane analog. Then we drew inspiration from ifosfamide and glufosinamide, which are alkylating agents that keep the cell from reproducing by damaging the cell's DNA. We were interested in incorporating this characteristic in one of our analogs as a method of inhibiting proliferation. And that's why we designed the 2-chloroethylurethane analog. Next, we designed the dodecyl and octadecyl urethane analogs. And as you can see, highlighted in pink are lipophilic tails. So we wanted to mimic the lipophilic structure of the ceramide in our analogs to see if this would increase the binding affinity of our compounds to acid ceramidase. We then decided to design the methoxyethyl and the ethyl isocyanoacetate urethane analogs to investigate the effect of various electron densities at the carbonyl on the binding affinity. And lastly, we designed the carbamate analog and the amide analog. And these two compounds have equivalent alkyl length as Carmofur, but they have different electrophilicity due to the single atom substitution that's highlighted in green. And we wanted to see how this would impact metabolic stability as well as Mpro and acid ceramidase inhibition. As we tried to come up with the best synthetic conditions for these analogs, we used 19F NMR for mechanistic insight. And as you can see here, we tested different bases for our different compounds, and different conditions resulted in various yields. So it was important for us to test different conditions because we want to find the best reaction rates and the best yields possible in order for the reactions to be more scalable and reproducible. And 19F NMR enabled us to do so because it showed us reaction conversion in real time. Using 19F NMR screening, we found that for a vast majority of the analogs, as the pKa of the base increased, reaction rate and percent yield also increased. Specifically, DBU had the highest pKa and produced the quickest conversion for most of the compounds. After we've synthesized all of the analogs, we decided to put them into biological assays to see how they would perform. So we performed multiple MTT assays in various cancer cell lines. And as I mentioned before, Carmofur was designed to treat colorectal cancer. So we performed an MTT assay with HCT116 cells, which is a colorectal cancer cell line. And as we can see from the IC50 values on the screen, actually all of the analogs have a lower IC50 value than Carmofur. And a lower IC50 indicates higher potency of the drug because it means that a smaller amount of drug is required to kill half the cells. And although the analogs did not show much activity in some other cell lines, when we tested them for 24 hours, when we did the MTT assay for 72 hours, it showed significant activity. And lastly, we evaluated the inhibition of Carmofur analogs against the SARS-CoV-2 main protease. All the compounds on the graph displayed some levels of inhibition of Mpro activity, and specifically the benzyl urethane analog and the hexyl carbamate analog actually performed better than Carmofur because it was able to inhibit Mpro activity the best. I would like to thank my group members, the alumni who worked on this project before, my advisor, Dr. New, Professor Downing for the high performance computer cluster, as well as an analysis for the 19F NMRs, the NMR instrumentation that we use. Thank you.